Hello, everyone. My name is Kevin Lance, and I'm the Director of Analytics Marketing here at Unchained Labs. I will be your moderator, and thank you for joining us today. We will have a Q&A session at the end of the presentation. So to ask questions, all you have to do is click on the Q&A button in the Zoom navigation bar at the top or bottom of your screen and type in your question. When submitting a question, please avoid clicking the anonymous button so that way we can reach out via email afterwards if we aren't able to get to your question during the webinar. We will get to as many of them as we can. And now I'd like to introduce Ali Shojai, Field Application Scientist for Particle Analysis at Unchained Labs. Today, Ali will talk us through how Hound can identify any particles, whether they are organic, inorganic, biologics, and even metallic particles. And now I'll hand it over to Ali. How are you doing today, Ali?
I'm very well. Thank you very much for the introduction, Kevin.
All right, great. Let's get going.
All right, so let's begin. And as Kevin mentioned, I'm going to talk about how we can use Hound for easy identification of mystery particles. As an application scientist in the particle analysis team of Unchained Lab, I'm very used to receiving calls from customers who suddenly start seeing visible and subvisible particles in their drug formulations. It could be in the drugs that are under development or the drugs that have been in production for a long time, and suddenly they start noticing particles in the formulation. And they're essentially trying to find solutions that can detect these particles and also provides an answer to what these particles are and where they are coming from. So they're trying to find the fingerprint of what these particles are. But since we are working with a Hound, they're trying to actually find the footprints of these particles. The issue is when we are working with formulations, we may have particles that come in many different forms. They might be just protein aggregates, or they might be the results of corrosion in the pipes. They could be leftovers from cleaning material, or residues from the APIs. They could also be coming from anywhere. They could come from caps, pipes, bioreactors, syringes, vials, anywhere. So these particles could be anything, and they could come from anywhere. So we need a solution that can handle any kind of particles, and that's exactly what Hound is. Hound is an instrument that is capable of characterizing every kind of particles. It's a combination of microscopy with 3 different separate systems of spectroscopy, along with image and signal processing capabilities. It is able to count, size, and shape the particles, and also provides chemical and elemental identification of these particles. Not only that, it is also able to handle all kinds of samples. You can filter out your solution and just look at the particles, or you can look at your particles as they are still in the buffer and essentially analyze it in the wet condition. So dry or wet doesn't matter. With the proprietary filters unique to the Hound, it is possible to analyze any kind of sample there is. So how does Hound does it? Let's start with microscopy. Hound microscopy system automatically detects, count, and size and shape particles. But one thing to keep in mind about microscopy is because we are using magnifications, we're usually looking at the very small area under the microscope. Hound is able to collect many different images all right next to each other, and then it will seamlessly stitch all of these images together and create one large image, which we refer to as a mosaic image, to detect as many particles as possible and analyze everything about those particles in terms of size, shape, and number of particles. Not only that, it will remember the location of every single particle that it has detected so that it can go back to it later and identify those particles. And if needed, it would also take pictures of every single particle. The microscopy system is capable of handling any particles from 2 micrometers all the way up to 15 millimeters. So once we create this gigantic image and identify all the particles, and we know exactly where each particle is, we can identify those with one of the spectroscopy systems that are built into the Hound. Hound comes with a Raman spectroscopy capability with 2 excitation wavelengths, both 785 and 532 nanometer laser sources that can measure particles down to 2 micrometers. So if you have a filament that you see under the microscope and you are wondering whether this is a protein filament or just some residual from the lab wipes, Raman spectroscopy can give you the answer that, oh, this is just coming from your lab wipes. Or if you have a strange particle and you are wondering if this is a piece of shattered glass or a protein, again, Raman spectroscopy will tell you that the particle is actually protein aggregates. There is a large database of over 150 individual materials that Hound uses to identify these particles. Customizing this database is quite straightforward and easy. And Raman spectroscopy is able to handle any kind of organic and inorganic material, including biologics and proteins. But as powerful as Raman spectroscopy is in terms of identifying particles, it has a big problem. Metallic materials cannot be detected with Raman spectroscopy. That is a big problem for Raman spectroscopy, not a problem for the Hound, because Hound also comes with LIBS, or Laser Induced Breakdown Spectroscopy, which is capable of performing elemental identification, including identification of metals and other inorganic materials. LIBS is capable of handling any particles down to 20 micrometers, and it also has a large database of over 50 unique compounds. And again, customizing this database is quite straightforward. So a combination of LIBS and 2 kinds of Raman spectroscopy gives you enough power to identify particles, any kind of particles that there is, using this databases. But that's not all that Hound does. You can tailor Hound to your very specific needs. So if you have proprietary materials or materials that are unique to your application, Hound is also work with those applications. I have one example of such material here. If, for example, we look at an aluminum cream caps on one sample with LIBS, we see from the particles a LIBS spectra that would look like the green spectra on the left. When we compare it to the reference spectra of aluminum in the reference database of the Hound, which is the blue spectrum on the left, we notice that they are very similar, except for one shoulder on the right side of the major aluminum pit, which is most probably the results of this aluminum being a specific alloy. We can add this specific alloy of aluminum to our database in a matter of minutes, so that the next time that we are analyzing an unknown particle, like the one in the left, instead of using pure aluminum as a reference, we're using this LIBS response from the specific alloy as the reference. So that way we would know whether this aluminum is coming from the caps or if it's coming from somewhere else. So it is and the whole process of adding this new reference to our library takes just a few minutes. We're going to talk about two real-life examples of how Hound helps solving problems. But before I do that, I'm going to answer one question that is very common with our customers, and that is, how does Hound know what the Raman or LIBS spectra is? How do you match the Raman response to your reference library? We do that through a process called matchranking, which is essentially just the linear correlation that correlates the spectra from the unknown particle to one of the reference spectra in the database. The process will give a score between 0 and 1,000. The higher the score, the more correlated, the more similar the materials are. This correlation number, this match rank number, depends on many parameters, but the most dominant factor is the fact that the peaks are present at the same location. For example, if we look at the spectra on the top, we notice that most of the peaks appear at the same location. So we get a very high match rank of 979. If we look at the spectra at the bottom, we notice that not all peaks are present at the same location between the reference and the spectra that we have collected. So we get a much lower match rank of around 553, and that way we know that this material is not the same as this reference library. So every time we have a particle, Hound automatically compares the spectra to every single spectrum that we have in our reference library until it finds the best match for that particle. That's how the identification is happening. With that being said, I'm going to start talking about the first case study that we performed, which was the study of the structural debris from a failure analysis study. So we had a customer who had some failure in their structure, and they had provided us with 3 different kinds of structural debris from the location of failure, and they wanted us to essentially identify what kind of materials were present in the debris. They also provided us with 3 different kinds of raw materials that they had used in constructing that structure that had failed. So we used those 3 different raw materials to create a custom database and then match the materials in the debris to those raw materials. We started by studying the raw materials. Interestingly, the first raw material that we studied was actually 2 different kind of material. It was either borosilicate glass or low alloy steel. The other 2 were dolomite and aluminum oxide. So we noticed that we had 4 different kinds of materials: glass, steel, alumina, and dolomite. And while Raman can easily identify borosilicate, glass, dolomite, and aluminum oxide, it cannot detect steel. So we used LIBS for this analysis. With that information and knowing what kind of raw materials we had, we looked at our 3 different mixtures of debris that we had: mixture 1, mixture 2, and mixture 3. And then we counted, sized, and shaped the particles and also identified them. In mixture 1, where we had around 1,700 individual particles, or 1,700 particles, we noticed that the majority of the debris was the steel particles, with some glasses and very small amount of dolomite and aluminum oxide. In the second mixture, we had aluminum oxide and low alloy steel with no glass or dolomite. And the third mixture was also dolomite, steel, and aluminum oxide, and no glass. This will provide a quantitative analysis of the kind of debris that we have, which would provide very useful information for the failure analysis of this study. For example, in mixture 2, we only have aluminum oxide and low alloy steel. That means that the failure has happened in part of the structure that is built from aluminum oxide and low alloy steel. And the failure was probably more severe in the parts that were made out of aluminum oxide. And then in mixture 2, the kind of failure had no effect on the parts that were built from glass or dolomite. So not only we were able to identify complex raw materials and structural debris, we were able to quantify the distribution of materials in our mixtures and also demonstrate the possible sources of failure from material based on just on the particles that we saw in the debris. So we got all that information from the Hound in this case study. With that being said, let's go to the second case study, which is a little bit different from the first one, which is about the protein aggregation study in biologics. A lot of you are familiar with polysorbate 20 or PS20, which is used very commonly to stabilize monoclonal antibiotics. The problem with using PS20 is that it would lead to the formation of oleic acid, or as I refer to going forward, OA, either as a result of degradation or raw material byproducts. The presence of OA may lead to the formation of protein aggregates. And if we look at it, we can easily see the aggregates forming as a result of OA. But what are these aggregates? We want to know if OA actually lead to aggregates in one real life problem, and if it does, what kind of protein aggregates are formed. So we had a customer who provided us with 3 different samples in 3 different buffers, buffer with low OA content, with moderate OA content, and high OA content. We have the reference Raman spectra of the OA and the protein, but we want to go deeper. We want to know how much OA we have in our protein aggregates. So we want to have essentially a spectra for each unique composition of OA and protein aggregates. And that's where the customizability of Hound will be very useful. We created a custom database that went all the way from 0% OA, 100% protein, to 100% OA, 0% protein in 1% interval. So we get a total of 101 individual spectra in one customized database. And then I'm not going to show all 101, but a few examples of this customized calculated spectra are shown here. And now that we have this customized spectra with different levels of OA and protein aggregates in it, we can go towards 3 different samples, 3 samples in 3 different buffers, and look at each of the aggregates. For example, if I'm looking at this unknown aggregates that I'm using as an example, I can compare it to all 101 different samples with different OA concentrations. And then look at that match rank number that I get for this specific example. For example, for this specific example, the highest match rank belongs to the composition of 13% OA and 87% protein. So this aggregate is 13% OA and 87% protein. We can repeat that for all other particles in our sample as well and use this custom database to essentially get insight into the ratio of the OA protein in protein aggregates. And that is exactly what we did. We studied the buffers and first we counted the number of aggregates that we saw, and after that we selected 50 particles larger than 5 micrometers and analyzed them for the OA protein ratio. First, do OAs actually lead to higher aggregations? While both low and moderate OAs had roughly the same amount of particles in them, between 1700 and 2000 particles in around 50 microliters, high OA content buffer had around 4,100 particles, almost double the low OA buffer, which is indication that high OA content leads to higher aggregation. But we did not stop there. We also looked at that based on the methodology that is very easy to develop in Hound, and Hound is actually built for developing these methodologies. We know that the average or the range of the OA content in our protein aggregates varies from 0 to 3% in low OA content. But if we go to high OA content, the OA content ranges from 0 to 14%. And for the particles that actually have OA in them, the average OA content jumps from 2.4% in low OA content to almost 9% in high OA content. And also, the number of particles that had OA or oleic acid in them goes from 10% in low OA content to over 86% in high OA content. So not only high OA concentration leads to more aggregation, it leads to formation of protein aggregates with higher OA concentration in those aggregates. So we get insight into both what these particles are, and we also gain insight into the chemistry of these aggregates, these protein aggregates that we have. So as a quick summary, we had this protein-oleic aggregates with unknown ratios of the two. We created or we trained our Hound database with predefined OA protein ratios and used that custom database to study our aggregates. The results indicated that more OA in buffer leads to more aggregates. It also indicated that the buffers higher in OA have aggregates richer in OA. And we were able not only to show that qualitatively, we were able to quantify the level of OA in the protein aggregates. So maybe we can even use Hound not only just for particle identification and identifying the source of aggregate, but maybe we can use that to optimize our buffer. Use this information about the OA content in the aggregate to strike a balance between the stability and aggregation. So Hound can do all of those can provide all those different informations for us. So in summary, Hound can provide a complete particle analysis for any application. It has flexible sample size, can analyze sample both in dry and wet forms. The two case studies that I showed, the first one with the failure analysis was done on dry particles. The second one was done on while the particles were still in the buffer. It is able to automatically count, size, and shape the particles, and it is able to create large mosaic images from microscopy images that it has, and then use those images to detect and identify particles in the samples. For the customers who need to be compliant with the 21 CFR Part 11 regulations, Hound is compliant with this specific regulations. All in all, Hound has everything about particle analysis covered. So next time that you notice particles in your samples, you can relax with the Hound. Hound can tell you what those particles are, where they are coming from, and it also can do it as fast as possible. So maybe, just maybe, because you have the capabilities of the Hound, you can take, when you see those particles, you can still take that well-deserved vacation because you're able to solve the mystery of what they are and where they are coming from. With that, I'm going to finish this presentation and thank you for listening to my presentation and hand it back to Kevin for any questions and answers that there are. Thank you very much for listening to my presentation.
Great. Thanks, Ali, for taking us through how to use Hound to address particle detection and characterization needs across all those different material types, and how to identify unknown organic, inorganic, and metallic particles, and also showing it can provide insights not just into particle identifications, but the details of the particles present in that sample, and it preserves vacation time too. Great. We have some great questions that have already been submitted. You can still ask a question by entering it into the Q&A section of your Zoom navigation bar, but let's start tackling those. So is it possible to, let's see, actually, let's go with this one. Why does Raman spectroscopy user need 2 lasers in the Hound?
All right, that's a great question, and we usually get that. So different materials have different ways of reacting to excitation wavelengths. Some materials have a stronger Raman response to 785 nanometer laser, for example, inorganic materials. Other materials, like proteins, have a much stronger signal when we are using a 532 nanometer lasers. So a combination of these 2 excitation sources ensures that we can identify all sorts of particles. In addition, sometimes with one of these excitation wavelengths, we get fluorescence that might be so strong that it just drowns the Raman signal, drowns the Raman peaks, and we can't identify the particles. Having 2 different sources help us overcome that problem in many different cases.
While the system is imaging automatically, how do you make sure it stays in focus?
Okay, that is the result of having a filter round that is unique to Hound. It's proprietary and unique to Hound, that when it sits under the Hound microscope, it gets stretches out, so it lies as flat as possible. That way, when we are moving around, because of this unique mechanical design of the sample holder, anywhere we go, because it's a flat surface, we are going to stay in focus and we are able to identify particles without worrying too much about refocusing every time that we're essentially moving around the sample.
Okay. And then I'm going to combine 2 questions here. So how does Hound direct particle identification to the particles and not the substrate or filter? And then the 2 questions that I'm going to combine will be, that's both for the dry filter and the wet round, where you could have background signals from the liquid. Okay,
so the filter rounds, both dry and wet, are gold coated, which means that they are not Raman active. We talked about the fact that metals are not detectable by Raman. The fact that we have a filter that is coated with gold means that whatever signal we are getting, it's just coming from the particles. So even if you have a particle that is transparent to the laser source of the Raman spectroscopy and the light goes through and hits the substrate, because the substrate is gold and non-Raman active, no signal would come from it. So everything that we see would be just from the particles. And then actually for the Hound, we have a proprietary nitrocellulose or CN filters that are not LIBS active. So again, any signal that we get is just from the particle itself. So we have the fact that we have this unique substrates or filter rounds for depositing particles help us to make sure that the signal is just from the particles and not the substrate.
And is there any concerns about that when looking at a liquid round as well?
No. The same thing applies if they're gold coated. I mean, we have a glass slide that is sitting on top of the sample, but glass has such a weak Raman signal that is not a source of concern for us.
Okay. Another question, we are only interested in fibrous materials in our samples. Is there a way to just ID those?
Yes. So when we are doing the identification, we can select to identify all the particles, which is not always practical, or define a specific criteria for which particles we want to identify. For example, identify the 100 largest particles, or select 10 particles from each size range to identify. We can also use the shape to select what kind of particles we want to identify. For example, only identify particles that have a fibrous shape. So ignore everything that is circular and only identify particles that have a fibrous shape. So because we have that capability built into the Hound, you can also identify the fibrous shaped particles without spending time on any other kind of particle that may exist in your sample.
Okay. Can you talk a little bit about your experience with Hound with samples that may have aggregated proteins in them, and the details of the question focus a little bit on looking at how many aggregates they are and how the size of the aggregates as well?
Yes, so essentially so Hound is built for essentially larger aggregates, so to speak. Anything larger than 2 micrometer can be identified with the Hound system. We've studied many different systems that had protein aggregates in them. We just talked about one example that we had done it while the samples were in the buffers. We have done it also successfully on particles that where the aggregates were filtered out, and we just studied the dried protein aggregates using the Hound. So we have actually done both of them. Depending on how much aggregates there is, we need different volumes of the samples. The example that I showed, we used around 50 microliter. We may need more or less than this volume, again, depending on how much aggregate there is and what size range of protein aggregates we are interested in.
All right, and I noticed we're just past the bottom of the hour, a little bit over time. So I think I'll make that our last question, and I'll say thanks for answering all those great questions, Ali, and thank you for a great presentation. I also want to thank all of you who joined us live today. For those of you that we didn't have a chance to get to your question, we'll follow up by email afterwards. And if you want to have a deeper conversation with our team, you can get in touch with us at info@unchainedlabs.com or through our website at unchainedlabs.com. We'd love to connect with you and talk about science. Thanks for attending our virtual seminar, and I hope you have a great rest of your day.
Thank you.